Good, thank you. Yes, I'm not a hydrologist. I will talk to hydrologists if I have to on occasion, and sometimes it's enjoyable. But I'm a plant ecologist and forest ecologist, and I'm going to be speaking more from the vegetation perspective and the project that Aldous and Axel and I were involved in and that Amy already presented on. So my co-authors is really Julie should be giving this talk because almost all of these slides are hers that she made, and I just stole them and then she explained to me what they meant, and now I'm going to talk about them. And then Anne McIntosh is the other co-author. So Anne was a PhD student with me, and she did the first part of the vegetation work at this study site, and she's now graduated, and she's an assistant professor at Augustana campus at U of A. And then Laurie Schrader is my technician that did a lot of the data collection on this project. So I'm going to talk about understory vegetation and a little bit of soil, nutrient, and water component in response to simulated mountain pine beetle. So we all know that mountain pine beetle broke out of BC and came into Alberta, and that it's a relatively unique disturbance in Alberta's forests. And the things from a vegetation response perspective that we're interested in is how it differs from fire, because we know these lodgepole pine stands are adapted to regenerate after fire. So the main differences are that the overstory is selectively killed, that the needles are retained for 3 to 5 years, so it's a slow death process as compared to fire, and the understory and the soil are not directly affected in any way. So all of these are dramatically contrasting with the disturbance that these forests are adapted to and what that they experience when they have fire. So we go through these stages, the green attack, red attack, and gray attack stage, and it's a relatively slow death process. And this is expected to influence the way that the forest responds to this, particularly the way that the understory responds, because the canopy and potentially the soil, water, and nutrients are changing slowly over time, as opposed to dramatically as they would with fire or as they would with harvesting. So our questions were how do understory vegetation and soils respond to mountain pine beetle attack, and then related to the hydrology component of the project, really what the implications might be for stand and watershed scale hydrology. So I'll just reintroduce the site here a little bit, just in case anybody has joined us since Amy's talk this morning. The site is located near Robb, Alberta, as shown in this map here. And we did this as a preemptive strike with stem injection of herbicide, stem injection of glyphosate to chemically girdle the trees and to kill them in a manner that would simulate really the way that the trees would die following a mountain pine beetle attack. And this allowed us to put in place experimental controls so we could have pre-treatment or pre-disturbance data collection, and then have an untreated control, 2 different levels of simulated mountain pine beetle attack, and then a clear cut that is meant to emulate what it would look like with the salvage logging. So we have a little picture here that shows the stem injection, and it puts these little 22 capsules that had glyphosate in them, and we girdled the tree with a number of these, and they did in fact die slowly over time in a fashion that quite resembles the slow death following mountain pine beetle. And this is the crew. It was quite the job to inject all those trees, I have to say. And we collected data in 2008 prior to treatment, and then had the treatment was in June of 2009, and we've been following it since. So we have 3 replicates of all our treatments for the vegetation component. So we have the hydrology plots here that Amy talked about, and where they have all the intensive hydrology instrumentation. But then for the vegetation component, we wanted to have actual true replications, and we can because we don't need $1 million worth of hydrology equipment for our experiment. So we had another set of blocks here and another set of blocks here. So we had 3 true replicates of all the different treatments, and then it's a Backy before-after treatment controlled by 3. They were all pure pine stands, 120 years old, sort of an eco site. Site index 22 to 24 meters in height, and we've been looking at it up to most recently 2016, collecting data at these sites. Okay, so in all of them we have a setup like this. In the entire treated block is divided into squares like this, And we have larger squares where we've been measuring downed wood and the response of the canopy, and then small plots within that where we've been looking at vegetation response. Okay, so we measured vegetation and ground cover in little plots like this, and then also looked at dead woody material, did some surface soil moisture measurements, looked at soil nutrients using plant root simulator probes, and then looked at forest structure and canopy cover doing hemispherical photos. So these are photos that you take with a 180 fisheye lens, and then you can import that digital image into some computer software that we use the program that Phil Como developed, and it gives you various metrics, and we just used it to calculate canopy cover. So it's a quantification of canopy cover. Okay, so I'll share the initial results. So this is the phase of the project that was Anne McIntosh's PhD thesis. So she collected the data in the pre-harvest period, so 2008, 2009, 2010. So just the very early red attack response, and we published this in Ecosphere in 2013. And so we found out that the treatments worked, that they did kill the trees, so basal area was reduced and dead woody material began to accumulate slowly. Most of the trees, dead trees, were still standing at this stage. Understory richness and cover increased very slightly in the mountain pine beetle treatment and declined in the salvage harvest treatment. Composition changed slightly, mostly in the salvage logged treatment. And the soil microbial communities and nutrients and soil water, there were very minor effects on those in this early red attack stage. So as we transitioned, so then we had a little quiet phase where we just did a bit of monitoring, and then we ramped up again when Julie came on board to do her master's and to follow up this work. And we wanted to look at what happens to these stands as they transition to the gray attack phase. And what we were expecting was that we would get an increase in understory richness and cover with the disturbance intensity as the trees died slowly and began to lose their needles and lose their small branches and whatnot. The canopy would open up more, and we would get the understory vegetation responding to that, to the increased light, and potentially to increased below ground resources, water and nutrients. And we expected to get a change in composition responding to that, with shade intolerant species responding more than shade tolerant species, and we expected to get an increased soil moisture and nutrients as well, because the trees were dying and not everything would be taken up by them. So that's what we expected. And so now I'm going to share some of the results. So in all cases we're comparing these 4 treatments. So we have the untreated control versus the 50% kill, which ended up being, I guess it was fairly close to 50%, maybe not quite 50% kill, and our 100% kill, which was not quite 100% kill, but closely, and then compared to the salvage logged. So we have a gradient of disturbance intensity. Okay, so I'm going to show a few graphs that look like this, and so I will explain what these letters are first here to get it clear before I start to go into it in detail. So we have some kind of response variable here, and in this case we're just comparing the pre-harvest to the most recent post-harvest, or the pre-treatment to the most recent post-treatment time period, so 2008 versus 2016. And then along here we have our treatments. In this case we have our untreated control and the 2 mountain pine beetle treatments, and these are tree responses, so we don't have salvage logged because the trees are all gone, so there's nothing to measure here. And so we can see pre versus post here, that's that difference here, and these letters are showing those differences. And then along here we see letters that show differences between the treatments within a given year. So in this case there's no letters below these because there was no differences between these 3 treatments in the pre-treatment period, which was reassuring. And then the differences between the treatments post-treatment are A, and this one was in the middle, and it was significant. Then B, A and B were significantly different from one another. So if you're disturbed by the letters, that can give you some orientation, and I'll try and emphasize what the main differences were. So we'll get rid of those and show what happened here. So this is healthy trees, declining trees, and dead trees, and 2008 here, so no differences between any of the treatments prior to treatment, so that's what we expected. And then post-treatment, not surprisingly, the live basal area decreased. with the level of MPB treatment, so 100% kill, much lower live basal area than the 50% than the control. And same with declining trees. The number of declining trees is shown here with the differences, and then here is the dead trees here. So more dead trees in the 100% kill than in the 50% or the control. So the treatment was doing what we expected. And this shows what the canopy cover was doing. So canopy cover here and how it changed over time. So the control here, it's bouncing around a bit. It did drop a bit over time. We did get some blowdown. There was a lot of clear cuts in the area, and we did get some blowdown, and there's some other natural mortality that you'd get in these stands over time, but more or less was stable. And then the 50% kill went like this, and we're still trying to figure out what the heck is happening there, why it jumped up and then went down. And then the 100% just kept declining over time. So we got a fairly dramatic decline in canopy cover up to 2016 after the treatment. But you see, it's still nowhere near zero. So the standing dead trees still do play a significant role in terms of shading in the understory and serving as canopy cover. It's just not foliage anymore at the gray attack stage. It's the stems and the branches. Okay, soil moisture results. So this is just very surface soil moisture. So Amy presented sort of below the rooting zone soil moisture and showed that it increased with the mountain pine beetle treatments. We were measuring right in the rooting zone. And the differences aren't that huge, but you'll see that the 100% kill and the salvage log here always were higher than the control. There's a lot of variation over time during the season with what was happening with the precipitation. So if we check any of these time periods individually, there isn't a significant difference, but still there is a trend overall for the treated stands to have slightly higher soil moisture. And I think probably the difference between what we see here and what Amy saw is that we're looking at the rooting zone, so the moisture that's in the rooting zone may be taken up by the vegetation, and what's not taken up by the vegetation is draining down relatively quickly, and so then Amy would see it as that increased soil water below the rooting zone. Litter cover here tended to be higher in the treated, and especially in the salvage the litter cover was higher, and that was a lot of fine woody debris that was associated with the harvesting treatment itself. But the other mortality that was associated with these other treatments also did result in higher litter cover, and again that was a lot of the foliage coming down, and by the end of the time period even some fine branches and ends of branches and whatnot were coming off the trees and contributing to the litter. Okay, in terms of changes to nutrients, we tested all the nutrients individually between the treatments in the different time periods and didn't get any significant differences. So we took an approach to look at an ordination and how they changed over time and how the suite of nutrients changed over time, and that's what we see here. So Aldous did show, I think, an ordination diagram. So I think you're probably familiar with these diagrams, but we have different years here, which are the different colors of the symbols, and the treatments are different shapes of the symbols. And symbols that are close together mean they're more similar in terms of the suite of nutrients and the amount of those nutrients that there were. And when they're far apart from one another, it means they were quite different. So here we have the control in 2016, and here we have a 100%. Here we have a 100% kill in 2016, and you can see different than one another they were. But this diagram I think might be a little more helpful in terms of seeing where the differences were. So the major differences were in the salvage logged treatment. So here we have the pre-salvage suite of values are in this circle here, and then the early post-treatment are over here. And we did get sort of some evidence of an Assard effect or a flush of nutrients right after the harvesting, and then it was relatively short-lived. And then here we have 5 to 7 years post-treatment is back over here. It's still different than the control, but actually more similar to the control than it was in the really short time period. And so for the other, the simulated mountain pine beetle treatments, there really was no dramatic differences in terms of the soil nutrient availability. So either there weren't a lot of soil nutrients mobilized as a result of the disturbance. So we were expecting some to be mobilized because potentially there'd be more light to the understory, so it would warm up, maybe a bit moisture, and so decomposition would kick in and you'd get a bit of release of nutrients. So either that wasn't happening, or the extent to which it was happening was completely compensated by any additional uptake by the vegetation. So in the end, we didn't see a lot of differences in terms of nutrient availability in response to the mountain pine beetle treatments. Okay, so let's look at the vegetation responses now. And we have another diagram that reminds you what the letters are. So here we have the treatments here across the top. So each graph is one of the treatments: control 50%, 100%, and salvaged here. And again, these letters are between years within the treatment. So we have this is how the treatment changed over time. And then these letters you'd have to compare from there to there to there to there, or in this case, compare here. So that's the control in 2016 to the 50% in 2016 to the 100% in 2016 to the salvage in 2016. So that's what shows those differences. So we're interested in both how they changed within a treatment over time, and in particular how the treatments differed from one another within any one given year. So what we see in terms of this is number of species, so understory vegetation species richness. It tended to increase in the salvage and decrease in the salvage and then increase. So you can see over time after the salvage logging, it dropped down. So we were losing some species in response to that disturbance, and then it increased and actually exceeded what it was prior to the harvest here. So we get this kind of a pattern here. And then in the tended to increase in the 100% mountain pine beetle treatment. So we can see that here. Going from the control to the 50% to the 100%. So technically with that AB, it's not significantly different than these two, but you can see it is intermediate between them and the salvage, which is different than those two. So it did increase slightly in response to the 100% kill, and that's probably attributable to some shade intolerant species that were able to come in as the canopy opened up with that 100% kill treatment. Okay, in terms of total vegetation cover, so this is cover of all the understory species that we looked at. It decreased in the salvage and then recovered. So again, you see this decrease in response to the salvaging disturbance. It's quite dramatic, and then it shot back up by 2014, 2016, and basically was the same cover, not the same species we'll see in a minute, but the same cover as prior to the harvest. And then in terms of the other mountain pine beetle effects, they went up and down over between years, but there was no differences between those treatments in total understory cover. Grass cover increased with salvage in particular. So you can see this big peak of grass cover, particularly in the two most recent years in the salvage logging. So we got a lot of grass coming in to those areas that were harvested. And in 2016, it was 3.5 times higher in salvage than in controls. And we also saw an increase in the 100% kill here. So you can see the grass cover coming up in the 100% kill. And again, it shows this pattern of going from here to here to here to here. So the 100% kill is in between the salvage and the 50% kill treatment. Forb cover decreased with salvage and then went up again. So same kind of pattern, didn't go up as much as the grasses, but it went down and then recovered. And then the forb cover didn't show any response to the other mountain pine beetle treatments. And then finally moss cover here. So it decreased with disturbance level, but it's recovering. So here we have the moss cover went down in the salvage, and now it's on its way back up, not quite where it was prior to the salvage, but heading there. In the 100% kill, the moss cover did decline relatively dramatically in 2014 and 2016, so our most recent measurements. So it went down, and even in the 50% here, we see it went down a little bit, although it seems to have been changing over time in this area as well. And the moss cover can change over time as a result of changes in moisture precipitation in a given year as well. So, okay. Oh, the moss cover wasn't finally. Shrub cover is finally. Okay, so shrub cover decreased and then recovered in salvage as well. So you see same kind of pattern here with it declining after the salvage logging and then going back up again. And then it increased in the 100% kill here, and then it didn't respond to the 50% kill. So hopefully what you saw from all of that is, although overall vegetation cover wasn't changing in response to the mountain pine beetle treatments, that different plant groups were responding somewhat differently to that treatment. And we see that in this analysis, which shows the composition of the understory community. So again, this is an ordination diagram, and the data into this is every single species that was in the understory and their relative abundance. So it's the species assemblage, and it tells you whether it was similar or different than the control and how it changed over time. So in 2008, we see here they're all overlapping with one another, all the different treatments, which we expect. This is before we treated any of them. And here we can see in 2016, they're all nicely separated from one another. This is the salvage, so it's the most different from the other. And then we have the 100% kill here, which is more different than the control, and the 50%, which are more similar to one another. So we really see a change in the understory assemblage of species that follows that disturbance gradient quite nicely. So the composition of the community is changing, even though the total cover was not changing that dramatically, or the richness wasn't changing that dramatically. So this is a diagram that also illustrates that. So this is the individual plots, our 3 replicate plots of each treatment, and the arrows show them over time. So these black arrows show the change over time in a given compartment or a given treatment area in terms of its assemblage. So we can see this black arrow here, and it went here, and then back over there. This one went there, 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 and this one went there, there, there. So you can see how much they're changing over time in comparison to these green arrows, which are the controls. So the green arrows, this one's changing a bit, but really they're changing relatively little over this time period, certainly in comparison to the salvage logging. So the salvage logging affected the composition the most. And then here we have that comparison between the green again being the control versus the 100% kill. So the 100% kill isn't as much as the salvage logging. These are all the salvage logging treatments over here. And then we can see the same comparison with the 50%. So that just illustrates the actual change over time that accounts for those differences in terms of the assemblage and how the assemblage is changing in relation to the gradient of disturbance from 50% kill to 100% kill to the salvaging. Okay, and this just shows a little bit about what characterizes the different treatments. So again, these are all the salvage logging treatments over here, and they were characterized by a lot of wood cover, a lot of finer woody debris on those sites, and grasses. And then these are all the control and the mountain pine beetle treatment stands, and they had, a deeper LFH layer, litter cover, other characteristics, and mosses and understory plant species that are associated with continuous forest cover, and those were maintained on those sites. And this shows some of the species. So here we have pine and elemus and Calamagrostis species here, and fireweed here are associated with the salvage logging, and then we get alder and vaccinium and mosses and Linnea borealis and Cornus canadensis, and those species here associated with the stands where the canopy cover was maintained, at least to some extent. So the final analysis that we did, or that Julie did looking at this, is an indicator species analysis. So this is an analysis that tells us which species are associated with which treatments. And we can see that the salvage logging had a number of species that were associated with it. So it was the most different from the other treatments. So it had the most indicator species. So it had, that's not a typo. They changed the name of Elamus to Lamus, which is like really annoying because it looks like it's a typo, but that's its actual name. Now Lamus innovatus and fireweed and Polytrichum, sort of an early successional weedy moss. And pine seedlings, obviously associated with salvage logging, and then with the 100% treatment, some understory species, and then with the control, interesting, it was the feather mosses that were an indicator of the control, and that was reflected in the fact that the moss cover was reduced in those treatments, the mountain pine beetle treatment stands, and definitely in the salvage logging. Okay, so summarizing this in terms of canopy cover, in the salvage logging it was gone and it went down in the 100% kill. In terms of moisture, there was some indication of it going up in 100% kill and in the salvage logging here, and then didn't change much in the 50% kill. In terms of vegetation cover, it didn't change in the mountain pine beetle treatments, and in the salvage logging it went down and then up again over time, recovered. And then in terms of vegetation richness, same thing happened, it went down and then back up in the salvage logging treatment, and it went up a little bit in the 100% kill. Okay, in terms of the different plant species, in terms of the shrubs, they went up in the 100% kill, and then in the salvage logging they went down and then back up again, not as much as they had gone down. Grasses went up slightly in all of these, and particularly in the salvage logging, the grass cover went up, which I don't think would surprise anybody in the room. Mosses tended to go down, up and then down, up and then down, and then in the 100% kill they went down, and then in the salvage logging they went down and then back up, but not to where they were. And then forbs went down in the salvage logging but weren't affected by the mountain pine beetle treatments. Okay, so conclusions from this, the greatest effects were for salvage logging, which wasn't really surprising. It's a much more dramatic and immediate disturbance than death by stem injection of glyphosate or death by mountain pine beetle. But in terms of the mountain pine beetle treatment that we had with increasing intensity, richness, understory richness increased slightly, total cover didn't change much, grass and shrub cover increased, moss cover decreased, and the composition changed. So the understory wasn't changing dramatically to the mountain pine beetle treatments, but it was changing subtly in terms of the composition of species that were there and the relative cover of different groups. So my conclusion in terms of trying to link this to hydrology is that, we really didn't see much in terms of changes in understory cover. So from that, I would infer that whatever's happening in the understory isn't likely to play a big role in terms of moderating any kind of evapotranspiration responses. Now, we talked about this morning, Brooks had a comment that maybe on a much richer site where you'd get you might get much heavier cover of some of these earlier successional species, and you might get a more dramatic response than we had here, and that might play a more important role in terms of hydrologic recovery. But for this site type, which is a very common site type in the area, the understory cover didn't respond a lot and probably isn't going to play a big role in helping recover evapotranspiration. The vegetation changes were mainly in composition, and so the question is what the potential impacts on successional development might be. Will there, certainly if you're starting to get some grasses on this site, that presents a concern in terms of competition with small pine seedlings that you might hope would be regenerating on the site and their ability to develop, or if you get shrub cover coming on the site, then that would be a concern in terms of how these stands are going to go forward. And that's it. Many field assistants helped with this, and many funders were involved. So that's all, and I'd be happy to take questions.